Why?
Easy.
Okay. According to Miss Meredith, there's a spot in your head.
What?
Yeah, so Miss Meredith told me it was.
What is that?
Miss Meredith said it's like right here.
Okay, I know it's Friday. Listen up. Y'all, I know it's Friday, and I know it is last period. Y'all want to go, but this is your essay. Today is your essay. It's on the next test. Pay good attention. It's something that you can understand and get a good grasp on if you pay attention. But if you don't, you're not going to be able to get it from the reading. Okay? What? You're not going to be able to get it just from the reading. You have to pay attention here to be able to understand it.
Hit the lights, bro.
I'm not going to hit the lights because most of the stuff I'm doing today is going to be on the board. What we're going to understand today, and actually Monday, it's going to take me 2 days to do this section, is how to make a protein.
I don't know how to make a protein.
Remember this? A chain of amino acids?
Yes, sir.
That's a protein.
They're joined by peptides.
Joined by peptide bonds. And they will fold up. Once you get this chain of amino acids built, it will fold up into a protein. And that's what the code on the DNA is for. The DNA code tells how to make a protein. And so that's what you're going to see here today. Again, this starts on page 336. You should have your book open, page 336. Let me move this down a little bit.
Yeah. 339. You have it up in the 336.
Okay, I can already tell you guys are starting to make random comments. And like I said before, it won't be tolerated anymore. Alright.
Who just threw that at me?
Drew, could you take the shirt off your head?
No, but
It's really distracting.
I can't see.
It's distracting for people in the back. It's like a horse with blinders.
Alright. Okay, so what we're going to do is we're going to do 2 sections. We're going to take our DNA code and we're going to copy it. And that first part is called transcription. You may have heard before, you're transcribing notes, something like that. That's copying. And so we're going to copy our DNA code, and then we're going to actually take it out of the nucleus to the ribosome. And at the ribosome, we're going to make our protein. And that's the second half of this thing. It's called translation.
Wait, I thought all A's had to be paired with T's. There's some U's on there.
Ah, we're going to learn that today, yes.
Oh, that's confusing now.
So we're going to do transcription first and translation second, and that will lead to making our protein.
Magic.
Okay?
Magic.
And to get this done, we have a new type of nucleic acid called RNA. RNA is going to help us make a protein. RNA is a little bit different from DNA. And RNA is held in a location that we call the nucleolus in the cell. Do y'all remember the nucleolus?
I sure do. Nucleus. Like a mini nucleus.
Let me show it to you.
In the nucleus.
Now, I'm going to open up the AP presentation for this.
We should just take AP.
Nah, might as well.
Yeah.
Whoops, wait, that's not it.
Those computers can do that?
Here we go.
Yeah, baby, yeah!
Okay, here's a cell. Turn the lights out briefly for this. In a cell, here's the nucleus.
Yeah.
The DNA is scattered all about in the nucleus. It's called chromatin. And This big ball that you see inside the nucleus is called the nucleolus.
Forms ribosomes.
It does form ribosomes, and I'll show you how it does that. It holds what we call RNA. And I have that nucleolus right here. This big blue ball is that. And this DNA right here is laying all around here, around the nucleolus, inside the nucleus. And do you see all the little yellow dots? You know what those are?
Ribosomes.
Ribosomes. This is one of them. This red thing. This red thing is one of them. That's a ribosome. Okay?
Why do the ribosomes look smaller than the nucleolus in that, but they're bigger?
Because I have to make it big to show you actually what's going on inside a ribosome.
Yeah,
But in actuality, it's small.
You know what those look like? They look like the sickle cell anemia blood cells.
What do?
Yeah, they kind of. No, they don't. The ribosomes.
This ribosome here?
Yeah. No.
Yeah, it does a little bit.
Yes, it does a little bit of this. Just a little bit.
Here, you're sleeping.
No, you're stupid.
Guys, I don't know, you can't help yourself?
Not a valid question.
Okay. So, how does this work? Well, the first part called transcription, your book shows you here in this picture. I got to show it to you on the board, and then the picture will make some sense. Okay? What happens is a special enzyme. I got to turn the light on for this. You got to follow this. It's called RNA polymerase. It's like DNA polymerase a little bit, but we call it RNA polymerase. It comes in here. It grabs some of the DNA. Now remember, the DNA is a real long string. It's like a piece of fishing line. Remember I said that? And so the RNA polymerase is going to reach in here and kind of unwind some of the DNA. And it's going to take RNA nucleotides from the nucleolus, one by one, and attach them.
Click.
Click.
It's just like the same thing as
Click. It's kind of like the same thing, except a little bit different. I'll show you how it's different. Click.
Did the helicase still separate it?
Let me attach. No, helicase isn't involved in this process.
How to separate them?
The RNA polymerase both separates and adds the new nucleotide.
Gotcha.
Good questions. Click. Click.
Wish I heard when I closed everything.
Click. Now we have formed a strand of what we call RNA. An RNA strand. Each of these things is an RNA nucleotide. And RNA nucleotides are a little bit different from DNA nucleotides. One, they have ribose as the sugar instead of deoxyribose. That's why it's called RNA instead of DNA. Ribose instead of deoxyribose. Do you know? Can you guess the difference between deoxyribose and ribose?
There's no
Sophie?
2 in deoxyribose and 1 in
No.
ribose.
There's 2
Oxygen.
There's no oxygen in ribose.
Ribose has 1 less oxygen than. I'm sorry. Deoxyribose has 1 less oxygen than ribose. That's why it's deoxyribose. The second big difference is this.
Blue.
RNA has uracil, U, instead of thymine, T.
So U and
So U and A go together. So this DNA right here, you can't see it, but I'm showing you the DNA here, is TCAAGC. So the RNA up here, what goes with T?
U.
A. A goes with T.
G.
G goes with C.
U.
U instead of T. Usually T goes with A, but in RNA there is no T, it's U instead. So now we have A, G, U, U, G, C.
You just put a G on
Oh, I messed up.
That's wrong.
C,
and then G.
So the RNA strand forms a little bit different than the DNA would have. And that's one of the differences. You have U's instead of T's. And this AGU, UCG, that is code that will eventually make our protein, which I've lost. Oh, here it is. Here's our protein. This code will eventually end up making a protein. It happens over here on the ribosome. I'm going to put our protein here. This is what our final goal is. The RNA is then going to detach and make its way out of the nucleus.
Why?
And make its way to the ribosome.
Alrighty.
Now, do you see that we have copied the DNA? We've copied it. It's not an exact copy. It's kind of like a reverse copy. A G where there's a C, and a C where there's a G, and a U where there's an A, and an A where there's a T. It's a reverse copy. It's kind of like a photographic negative. Have you all taken photography?
Yes, sir.
In a negative, there's black where there was white, and there's white where there was black.
I'm doing that right now.
Well, this is like a photographic negative of the DNA. It's kind of reverse of what the DNA was, but it's still got all the information. It's still a copy of the code. And now this thing will then make its way out to the ribosome. And this is actually how ribosomes are made. Anthony, you hanging with me? Chuck, it looks like you're asleep. Listen and look. Okay? Sit up. It's always rude to be laying down when someone's trying to talk, especially when they're trying to teach you.
I'll have a shirt on their head.
Now,
It's not read at all.
you're making the comments, right? Yeah.
I'm just trying to design.
What if they had a shirt on their head? Is an example
I'll
of the
take
comments
one
I don't
after
want
that.
you to say. Do you see what I'm saying? I'm doing it without getting too angry. That's good, man.
Yes, I'm proud of you.
Okay, I'm working. I'm working. Will you work too?
I will work.
Try not to say stuff like that.
Yes, sir.
Here we go. How do you make a ribosome? Well, it's the same way. These things right here will go out here and form themselves into the shape of a ribosome. That's how ribosomes are made. That's why the nucleolus makes ribosomes. And by the way, it's hard for me to move these things around because they're separate. So I have made a long strand that's hooked together that I can move around easily.
That's some hard work. That's a funny word.
So this thing will come out of the nucleus and will actually shape itself into the form of a ribosome. That's what a ribosome is. It's RNA. And we call the ribosome, the ribosomal RNA, we call it rRNA. rRNA means ribosomal RNA. The ribosomes are made of RNA. And how did they get made? The way I just showed you. The DNA is unzipped, the nucleotides are put on one at a time, and then the RNA leaves and becomes a ribosome. Yes, Graham?
What are the RNAs attaching to make a ribosome?
There are some other proteins out here and stuff
Oh, okay.
that it attaches to. So it's actually RNA and proteins. That's a good question. Mm-hmm. Y'all see how ribosomes are made?
Yep.
Now, in this same way, proteins are going to be made. But it's a little more complicated. But this process of making a piece of RNA from your DNA and the RNA leaving, that's called transcription. That's what this is showing. Here's the DNA. It's been opened up. RNA nucleotides were added one at a time by this RNA polymerase enzyme. And then the whole thing leaves. Video footage?
Yes,
Yes.
please.
Okay, then you're going to ask me questions, okay?
Yeah.
To make sure you understand it. Ellen Grace, you getting all this?
Yep.
Are you excited about it?
Oh.
Very.
Oh, I missed the enable L. I'm sorry. I'm trying to work on that too.
I saw it.
Listen.
The cell transcribes the genetic designs for each protein from strands of DNA to special molecules of RNA.
That's RNA. RNA polymerase.
An enzyme unwinds a segment of DNA, making room for its complementary RNA nucleotides. Both DNA and RNA nucleotides contain a sugar, phosphate, and base. But in RNA, the sugar is ribose, and one of the 4 bases is replaced by uracil. The exposed bases of the DNA strand serve as a model for the construction of messenger, or mRNA. On other strands, the cell produces transfer, or tRNA, and ribosomal, or rRNA.
Look at it
In
fall
their
out.
final form, these molecules play a vital role in protein synthesis.
And now where's it heading?
To a ribosome.
To the ribosome, which is also made of RNA. Exciting, isn't it?
Very.
That's half.
Wait,
That's half your essay.
It goes to the ribosome or it goes to the nucleolus?
It goes. It starts in the nucleolus. These pieces do. They attach. They get in the right order. Then they leave. They can become a ribosome,
But where do they go?
or they can go to a ribosome to be made into a protein.
I got you.
Questions?
Do that again. I was making sure you did that. I was like, what?
You're hearing things again.
One more video footage of it. Enable all. I'm doing it.
That'll go sound.
Watch it.
Share this.
Transcription is the process by which RNA is assembled from a DNA template. The 2 strands of the DNA double helix are held together by bonds between complementary base pairs. DNA encodes genetic information in the sequence of bases along one strand. The portion of the DNA molecule that is a single gene or coding region is bounded by termination and promoter sites.
We didn't learn about those. Don't worry about the termination and promoter sites.
This is the worst one.
Now this is good. What?
A molecule of RNA polymerase binds to the promoter site. It moves along the DNA, separating the 2 strands of the double helix. Each now unpaired base will bind to a nucleotide in the vicinity which has the appropriate complementary base. In the synthesis of RNA, uracil is the nucleotide complementary to adenine.
See the piece of RNA falling off? Right there.
This process ceases when RNA polymerase reaches the termination site. The DNA strands bind to one another once again as the new RNA molecule moves away. This RNA is a copy or transcript of the message contained in the gene.
See the RNA there, and that's going to head out of the nucleus.
Fascinating.
Exciting, huh?
Very exciting.
Okay. Now here we go. Here's the other half of your essay. Oh, we already saw that. Let me show you what's going to happen here. The RNA, in this big long piece of RNA that's going to go into the ribosome, has a name. This piece of RNA is called mRNA.
Measure.
mRNA stands for messenger RNA. This is a messenger bringing code from the nucleus over to the ribosome. Messenger RNA. And the messenger RNA is read 3 letters at a time. Tucker, hang with me here. The first 3 letters that you see here are called AUG. I'm just reading the letters. Adenine, uracil, guanine, AUG. And those are the first 3 letters. We call 3 letters, we call that a codon.
Why?
That's just the word for 3 RNA letters. And a codon codes for a certain amino acid. AUG is code for the first amino acid in the protein.
Oh yeah.
Which you can read this chart to figure out what that amino acid is. Can you find AUG on the chart?
Yeah, it's the only one that's highlighted in blue.
It's methionine. That's the start codon. It's always the first amino acid in a protein. Every protein starts with methionine.
One says UGA stop.
Yeah, UGA is a stop codon. I'll tell you about that in a minute. So the next codon is CUC. Find CUC on the chart. And by the way, this chart is in your book on page 338. Find CUC.
Lysine.
Leucine. Leucine. Leucine will be the second amino acid. So in the protein that we're about to make, methionine is first, leucine is second. Or the way y'all are looking at it. Methionine is first, leucine is second. How about the third codon? CAC. Histidine. That will be third.
So wait, those are amino acids.
These are amino acids. A protein is a chain of amino acids. And the DNA codes for the order of the amino acids in the chain. Yes, Anthony?
Each amino acid has a different protein which makes up one big amino acid.
A bunch of amino acids together make one protein. A protein is a string of amino acids. Okay? Yes?
Do you know this chart like by heart?
I do not. I don't know. I've never heard of anyone who's memorized the chart. That would be cool, though.
Extra credit?
There's UGA up there.
Yeah, we'll talk about UGA. At the end, once your protein is done, once all the amino acids are strung together, and by the way, a normal protein might have 500 amino acids. But at the very end of the long string, the last codon codes for stop. That means stop, we're done with the protein. And I'll show you how it ends. And there are 3 stop codons, UAA, UAG, and UGA. I always remember UGA was a stop codon because I went to Georgia Tech. And I remember UGA, University of Georgia, stop. No, we don't like that. You see? So UGA is always easy for me to remember, stop codon.
What?
No. Stop.
Stop. No UGA.
How's she stop there?
UGA, University of Georgia? Okay, so.
Bro, what would the next ones be? I'm trying to get this how you got CAC.
Well, I'm just reading. I'm reading the code.
Oh, okay.
CAC from these nitrogen bases.
Okay,
You see what I'm saying? And where did that come from? That came from the DNA. So your code on your DNA will be transcribed into a piece of mRNA, which will go to the ribosome and be read 3 letters at a time to make your...
Do they always start with AUG?
It always starts with AUG.
So does that mean
every protein.
That's the mRNA chart?
This is the mRNA chart.
Okay, so if you have like the next one, say, like it's like, what's the next one?
And the next one is GGG.
Glycine, boom. Who's fast? Grammars. Got you.
So that means like, okay, got you. I was just trying to figure out how you were getting the
Yeah, it means glycine will be next. And then next, and then next, and pretty soon you'll have all your amino acids, and that will fold up into a protein.
Got it.
Okay? Now, we're not done.
Sure we are.
Now we got to see how these 3 letters are made into an amino acid. And that's where these guys come in, these green guys. Got to turn the light back on for this.
Are green guys bad guys?
I do. I got to turn the light back on.
Are those tRNA?
They're tRNAs.
Translator or
Transfer.
Hold on.
No, it's translation.
Now, this is everything that I'm about to show you is happening in this picture in your book. This picture is very confusing.
It is very.
If you weren't listening today, you would look at this picture and you would go, I don't understand. But I'm about to show you, and you're about to understand. And it's going to be good, because then you'll go off for the weekend and you'll go, I understand this stuff. And then you can party and have a good weekend. Party isn't like a birthday party. Not as in some kind of party the way y'all are thinking of.
I saw you driving yesterday.
A rave.
Texting and driving.
Hey, Sophie.
Which is illegal.
I wasn't texting and driving.
Obama.
It is not illegal.
Okay, listen.
It's illegal in all 50 states now.
Listen.
Is it really?
bond.
Listen to me. Are you ready?
Yeah.
These tRNAs are molecules that carry amino acids. And they're made of RNA too. Listen. These molecules here are made of RNA. They're made of this stuff. All coiled up into a weird shape.
Looks like a T.
And each of them carry an amino acid with them. Now, at the bottom of their structure is what we call an anticodon. The anticodon on the tRNA will try to match up with the codon of the mRNA inside the ribosome. Let me show you how this happens. Well, I'll leave this. Watch this. Watch. The tRNA floats into the ribosome. Here's our ribosome. The tRNA floats into it, and its anticodon tries to attach to this codon. Remember, these have certain shapes, and they can try to attach.
Nope.
And that doesn't fit, so that would leave. And then another one would come in and try to attach.
Nope.
And that doesn't fit either, so that would leave. And this process continues until one comes in and tries to attach.
Yepers.
It fits. The anticodon attached to the codon correctly. What is the anticodon? Can you guess what three bases are those that match with AUG?
Nitrogen fifth.
U.
U is correct. A is correct.
C.
C is correct. UAC matches with AUG.
You know what USC is?
UAC.
University of.
It's tyrosine.
Actually, it's University
This-
of Arizona.
This tRNA, listen, can you guess what amino acid this tRNA carries?
Graham does my hands up, that's fine.
It was on the chart.
It's
Meredith? No?
Okay, you were wrong.
It's tyrosine.
No?
Read right.
Because it's the codon that codes for the amino acid, not the anticodon.
my bad.
What does AUG code for?
Oh.
Methionine.
Methionine. Very good, Anthony. So check this out. AUG,
Isn't methionine the only way it can
To use the red
yes.
But it's hooked up to it, that's why.
AUG codes for a tRNA that carries methionine.
So one protein would look like that.
No, this is an amino acid.
One amino acid would look like that though?
This is one amino acid. See, this is a protein, it's a chain of amino acids.
You know what?
So that's one amino acid.
What?
I understand this.
Okay, well, we're not done.
Oh, good
We're not done. The next, inside the ribosome, the next codon is exposed. And here we go. Here we go. tRNA comes in, tries to attach, doesn't work. Another one comes in, tries to attach, doesn't work. Another one comes in, tries to attach.
It's
fits.
leucine.
It's leucine.
Leucine.
Okay. I got the wrong one here. I'll switch it. There, that's leucine. And now, we have two tRNAs inside the ribosome attached to the mRNA. Now these amino acids are side by side. The next thing that happens is a bond forms between the two amino acids.
Oh my goodness.
You know what kind of bond forms between two
Peptide
amino acids?
Peptide.
That's a peptide bond. This amino acid is now bonded to that one.
Ouch.
Listen, the first tRNA now leaves. And it's going to go get another methionine. There's one. Because that's the only thing it can carry. Methionine. And the whole ribosome will now move. Now the next codon is exposed. Another tRNA comes in, binds, another peptide bond is formed. This tRNA will now leave and go get another leucine, even though I don't have one up there, you could pretend. And the whole thing moves down. Look, we're making a protein. The next codon is exposed. This thing comes in and binds. Peptide bond. The first one leaves. Goes and gets another yellow. The whole thing moves down.
And it goes and gets.
This happens fast. 15 amino acids per second
Goodness gracious.
fit onto the chain.
I run 15 yards in a second. I run 16.
So you pretty much talking about it.
That's
Now this is going to continue, and I lost the next tRNA. I don't know where it is, so I'm going to pretend this fit. Another bond. Now wait, we're to the end. Imagine we're to the end. In actuality, this would be about 100 amino acids long or more. What's the last one?
mRNA.
Well, I'll tell you, you can't read it from where you're sitting, but this says UGA.
uh-oh.
Stop, no, Georgia, no. Okay, so what does that code for? That codes for the releasing factor. The releasing factor is a tRNA that comes in there. It bonds. Now there's not another amino acid to bond with. This tRNA will leave. Go and get another orange one if I had one. And it's not bonded to anything.
That's the whole.
There's your protein. It's floating out there in the cell. It's been made. We made one of these.
Yay.
That's it. What's going to happen is the stop factor will leave. The ribosome actually comes apart and opens up. The mRNA is free to go do another. It can go through another ribosome and make another of the same protein. And often, mRNAs will go through 40, 50, 60 times and make the same protein over and over again.
That's crazy. What happens to the mRNA after it's done?
Finally, at some point later, the cell will destroy the mRNA.
Oh.
Yay.
Video footage?
Yes,
Oh, yeah.
Absolutely.
No.
How many think they understand it? Some of the basics. We're going to go over it
Drew
again
does.
next time.
Drew does.
We're going to go over it again next time. And do this whole thing again one more time for you. But that's basically it. That's your essay.
Okay.
See, you won't understand it by just looking at this. That doesn't make any sense.
Do we just draw this on our essay or just write
Now you just have to write about it.
Right there.
Good question, Sophie.
Okay, watch it.
Translation is initiated by formation of an initiation complex consisting of the smaller ribosomal subunit, the first amino acid tRNA, and messenger RNA. The larger ribosomal subunit then joins the complex.
Yeah, those are.
Proteins called initiation factors are also involved, but are not shown. The 70S ribosome has two sites to which transfer RNA carrying amino acids can bind. One is called the peptidyl or P site,
We didn't
and the
talk
other
about
is
that.
called the acceptor or A
We
site.
didn't talk about that.
The initiating transfer RNA carrying its amino acid
Watch
binds
it.
to the P site. A transfer RNA that
I'll do the talking since. Okay, so here comes another tRNA carrying an amino acid. Then they'll form a peptide bond between them. There we go. Now they're bonded. Now this tRNA is going to leave. Bye, go away. The ribosome is moving down, and this tRNA takes off.
Remember the next
Another
one.
tRNA comes in. UAC is the next codon. And now they connect. Are you watching this?
Absolutely.
Alan Grace, you got this?
Got it.
There you go.
This one's going to leave.
Watch.
And watch, the whole thing will speed up. I think they get it going faster this next time.
Tucker run out.
Yeah, here we go. And this just happens over and over again. 15 amino acids per second.
And that makes that's the protein.
That's the
chain?
protein forming. That's the amino chain of amino acids.
So the protein is just like the DNA strand. It's just long stranded. It's not coiled or anything.
A protein is a long chain of amino acids that then coils up into a protein.
Oh, so that's the amino acid chain.
This is an amino acid chain.
And then when it condenses, it forms.
It'll condense into a protein.
Well, I think you explain better than videos sometimes.
Oh yeah.
The video's better.
That's why I'm here.
Because like certain videos are better. Like I like when you talk.
Why is it so quiet?
Oh wait, we don't want to watch this one. I got one more I want to show you. This has scary music.
Meredith really likes music. Probably gonna turn it up though.
This is it right here. You ready?
Ready?
Meredith.
What?
Eyes.
A DNA molecule provides a series of signals.
This is the whole thing. There's the DNA.
Production of protein. The letters of the code correspond to the bases along the double helix. C for cytosine, G for guanine, A for adenine, and T for thymine. Each protein molecule has its own code, determined by the sequence of these bases. Beginning at an active site, a group of enzymes binds to the DNA molecule. This enzyme group is able to read the coded message. Part of this group travels down one strand of the helix. It chemically recognizes the bases as it passes, and matches to them other nucleotides from the surroundings, creating a new molecule. This is done according to the correct pairing rules. C with G, and A with U, where U stands for uracil, which replaces thymine in this new molecule. The new molecule is ribonucleic acid, or RNA. RNA is an intermediate step in the production of protein.
It's going to the ribosome.
These molecules of RNA are called messenger RNA because they carry the transcribed message to another part of the cell outside the nucleus to be decoded.
There's the ribosome.
RNA travels to a ribosome. It is here
Oh
that
man.
proteins are actually manufactured.
Sounds like my nightmares at
The
night.
ribosome reads the message from the RNA
Wait, in
to another group.
Okay, y'all have a good weekend. We're gonna do this again, the same section on Monday.
Oh my goodness.
Yeah, we just did it.
Check this out.